Continuing the eukaryotic ultracellular structure, we're now studying the plant structure. And of course, as it has a nucleus containing a nucleolus, nuclear pore, chromatin, and a nuclear envelope, this definitely 100% approves it into the plant kingdom. And not only the plant kingdom, though, that have a nucleus, but also like the eukaryotes. But having the membrane-bound organelles is what it does all. Of course, we know some organelles have a membrane and others do not, like the centrioles, which are actually absent in the plant structure but present in the animal structure. For chloroplasts and amyloplasts, they are definitely found in some plant cell, but for the cell wall, for example, it yet is a plant cell, but it doesn't have any chloroplasts. And that's because it doesn't need them, like the epithelium or something. Other, like the root hair cells, all these don't have a chloroplast, while all of them share the cellulose cell wall. And that's the material of which it's made from, of all plant cells. But for the centrioles, they're absent, we just said that. And it also, as it's a eukaryotic cell, it contains both 70S and 80S ribosomes. For the 70S, they're found in the mitochondria and the chloroplasts. But for the 80S, it's found everywhere, lying here in the nucleus, you can find it. And in this rough endoplasmic reticulum surrounding the nucleus, you can find the ribosomes attached to it. Of course, they're lying freely in the cytoplasm or attached to the rough endoplasmic reticulum. It has such unique organelles such as the amyloplasts, and these are only found in the parts of the cells that store starch, such as potato tubers, for instance. Those ones must contain the amyloplasts. They look like something like that, having the starch grains all over their structure. But we're now going to draw a 2D shape of a plant structure that's easier for you to comprehend. Alright, let us draw it here. I'm going to draw it in a rectangular structure, just like the palisade one. Okay, now I gotta draw the cell wall. Like that, rectangular. I surely need to fix this. Okay, this was about the cell wall and the cell surface membrane. Inside we have lots of mysteries, but let's get started with the nucleus. A circular nuclear membrane, also known as a nuclear envelope. We have the nucleolus right here inside, the uncondensed and unfolded chromatin, and those are the nuclear pores. Okay? Let me move it here a little bit. Surrounded to that, we have the rough endoplasmic reticulum, of which we find all the foldings like that. And we have to attach some ribosomes to it. Okay, let us use this. All over its structure, we have to attach the ribosomes. And here you go. For the ribosomes, we can draw more of them everywhere in this cell here. Okay, now I gotta draw something really. We can also add some smooth endoplasmic reticulum right here, also attached to that, looking like fingers. I know. No, don't readjust yourself. It's tubular shaped and it's membrane connected. Also, that was for that smooth endoplasmic reticulum. Now let's draw the Golgi body, which of what I say, it's like a Wi-Fi sign. It's like that, and you keep decreasing. It's not membrane connected. Like that. And it keeps decreasing. You gotta draw whatever you want. And the vesicles are surrounding it. There are many. We have transport vesicles, secretory vesicles, the Golgi vesicles, which are also known as secretory vesicles. So these were the Golgi vesicles. What else do we have to draw? Of course, we cannot forget the chloroplasts in the mitochondria. Let us start with mitochondria here. Oh, that was pretty messed up. Okay, so this is a mitochondria. We of course don't have only one in one cell, so here we go. Another mitochondria right here. The chloroplasts, of course, which we cannot forget. We're studying them on a wider perspective on the next slide. These are the chloroplasts. And we can probably add the grana of chloroplasts in order to determine it. Okay, that was a chloroplasts. It's not going to be really visible here. Another chloroplast. The amyloplasts, which we can draw also, something like that. I have to draw them attached. Okay, that was about this. We gotta have some lysosomes lying everywhere too, which are these vesicles. And how could I forget the vacuole? So here is a vacuole of any shape. It's gonna increase in size, I gotta add my labels. So that was some adjustments. We now have the cell wall, cell surface membrane, Golgi vesicles, lysosome. We can say this is a tonoplast of the vacuole. This is the cell sap of the vacuole. And all these together give us the vacuole. That's a chloroplast, a mitochondria. A rough endoplasmic reticulum which has a cisterna. We can add this. Okay, this is a cisterna. And this is a ribosome. And a myeloplast. Alright, what else do we have? Of course, the nucleus. Nucleolus. Nuclear pore. Nuclear membrane. Chromatin. These are our labels. Smooth endoplasmic reticulum. Okay, let me add my labels now. And here you go. Now let's move to the chloroplast section of this class. As you must know, of course, chloroplasts play a major role in every plant cell. We're not gonna say every plant cell because we know it's absent in some of them. But we can say in every plant that has the green color, it helps it to survive, okay? Because it makes itself, but it makes its food by itself, okay? So for the chloroplasts, they have a grana of chloroplasts. By grana, I mean the stacks of thylakoids. What is a thylakoid anyway? Well, these stacks coiled together, they all make the grana. All this is a grana. Or actually this one, not all this, my bad. This is a grana, and each one of these is a thylakoid. Okay? And these contain the chlorophyll. So as you can see here, that is one thylakoid, and this is the whole stack, the grana. Well, granum actually. So if you're talking about this part, this is called granum. while the plural form of it is grana. This is the plural form, so take care. It has a biconvex shape, just like the mitochondria. Like if you're drawing the mitochondria, you're going to also draw it like that. And this is called a biconvex shape. Stroma is where enzyme catalyzed reactions take place. I'm going to say like this is like the cytoplasm. If you're talking about this stroma, I'm going to say this is cytoplasm of the chloroplast. We call it stroma. But if you can remember, for the mitochondria, if you're drawing a mitochondria, okay. For the mitochondria, the thing lying out inside it is called matrix. And it also represents the same thing as stroma. Here, this is no, the stroma. The stroma itself is the liquid or the jelly-like material that contains water inside it. Okay, so that was about it. It has chlorophyll, of course, that changes the light energy that we take from sunlight into chemical energy. How exactly chemical energy? By chemical energy that it uses. Like in the plants, they use the carbon dioxide of which they get from air with the water of which we water them to form the oxygen and the glucose, as you can see, plus the energy produced. We're not going to say energy produced, it's released. Okay, so please take care, do not say energy produced, just like I said. We say the energy is released because energy is neither produced nor wasted or died, as this is a rule that most of you are going to know, of course. So the energy is released that makes it make its food by itself. Okay, so that's why the light energy is changed into a chemical energy through photosynthesis process. Okay, also it has 70S ribosomes, of which we said it has an 18 nanometer size. Starch grains, which look like a lobe like that. This is the starch grain. A loop of DNA that is also a circular one, of which we discussed before that it doesn't involve itself in the mitosis or meiosis. Of course, it's not going to do that. Lying in this stroma. So these things are lying in the stroma. It has an intermembrane space. This is the thylakoid, outer membrane, and inner membrane, as it's a double membrane bound organelle. But let's look at this further in a 2D shape we're drawing ourselves. So for mitochondria, I'm going to draw the same thing I did for the chloroplast. It's the same thing as the mitochondria, I mean. It's this way. But I add a double membrane bound organelle, so you add an extra double layer. My hand is so shaky, my bad. What I'm adding now is the ribosomes that are everywhere. Okay, so these were the ribosomes. Now I must add the grana of the chloroplast. This is 1 granum, the stack of thylakoids attached together with another one. Stick together. You can also attach it one more time by these oval-shaped thylakoids. Okay, that's for these. We can have another ones too. Another one. We have our loop of DNA that's circular and lying outside. That's it. And also for this starch grain, they're lobed, but I'm gonna draw them right here as some circles. You cannot show the lobe shape, and they're not even viewed like that. These are for the starch grains. I gotta label my shape now. This is a starch grain. This is a stack, or not a stack, this is the thylakoid itself. But this whole thing is the stack of thylakoid called grana, a circular loop of DNA. Another one, of course, I'm not going to mention. And the 70S ribosome, that's the outer membrane, but let's not make it busy here. Like the outer membrane is that one, and this is the inner membrane. And of course, we have the stroma. And here we go. And that was for today's class. Thank you guys for watching.